Good morning, everyone. Thank you for joining us. It's just exactly 10:30, and I know we have a lot of people joining from their previous meeting on Friday morning. So we're gonna wait maybe a minute or two before we get started. So if you need to go make a quick run to refill your coffee, now is your chance. We ask that everyone keep their microphones muted during the talk to reduce background noise. But if you see a colleague in the chat and you want to say hi, please go ahead and do so. And we'll get started in a couple of minutes.
I'll say hi verbally. Hi, Priya.
Hi, Nevan.
How are you doing?
Good. How are you?
Good. Nice to see you.
Good to see you too. Next time we'll have to have you in person.
Yes, I know. I'll look forward to that. I like your artwork in the back.
Yeah, we have some good artists in the Shaw Lab visiting every now and then. Your whiteboard looks the same as ever.
So we have a few people still joining us from the waiting room, but we have about 45 people. So I think we can get started.
Okay, thanks, Ana Lucia. Let me roll the ball over here. My name is Prasant Mohapatra. I'm the Vice Chancellor for Research here at UC Davis. We started this series of meetings on Fridays probably about 18 months back, and it has continued with all of your interests for a long time. The name of the meeting morphed into a different title now on Teams Research Forum, so which truly reflect the goal of these meetings. So continuing with the series, today we have a guest speaker, and let me request Dean Brashier to introduce our guest speaker, and we will get started with the talk. Allison?
Great, thank you so much, Prasant. And I think we were just thinking that this was number 78, and we started these in April of 20. Our guest speaker today is Kevin Krogan, PhD. He's a molecular biologist at UC San Francisco, where he's the professor and director of the intensely interdisciplinary Quantitative Biosciences Institute under the UCSF School of Pharmacy. He's also a senior investigator at the Gladstone Institutes. He's led the work to create the SARS-CoV-2 interactome and assembled the QBI coronavirus research group, which includes hundreds of scientists from around the world. His research focuses on developing and using unbiased quantitative systems approaches to study a wide variety of diseases with the ultimate goal of developing new therapeutics. Nevan serves as the director of the Hark Center, an NIH-funded collaborative group that focuses on the structural characteristics of the HIV human protein complexes. He's also the co-director of the 3 cell mapping initiatives, the Cancer Cell Mapping Initiative, and the Host Map Initiative, and the Psychiatric Cell Map Initiative. All of these initiatives map the gene and protein networks in healthy and diseased cells, with these maps being used to better understand disease and novel therapies to fight them. He's authored over 250 fields of genetics and molecular biology, and he's given over 350 lectures around the world. He's a SEAL Scholar, a Keck Distinguished Scholar, and recently was awarded the Roddenberry Prize for Biomedical Research. It's really my pleasure to welcome him to this forum, and I think he's going to give us insights into targeted drug discoveries for SARS-CoV-2. Thank you so much, Nevan.
Thank you, Allison, for that generous introduction. I'm just going to share my screen, and Prasant and Ralph and Anna and everybody else who invited me here. Can you see my screen?
Yes, it's perfect. Thank you.
All right. So as Allison kind of alluded to, what I'm going to be talking about is some ongoing efforts focused on COVID-19 under the umbrella of the Quantitative Biosciences Institute and the QCRG, the QBI Coronavirus Research Group, which has involved many different groups here at UCSF and at QBI. Actually, this is a picture on the steps here of Genentech Hall that was taken, maybe a week before the shutdown in March 2020, one of the last times many of us were physically together. But this group actually then was augmented to include over 40 groups and hundreds of different scientists here at QBI and at UCSF. And the group got so large, we actually had to break it up into different subgroups focused on different biological processes the virus is hijacking during the course of infection, or different technologies that we're using to study the virus and how it infects our cells. I'm going to be focusing on one of these subgroups later, the one that is led by Oren Rosenberg here, and Clem Verba, a QBI fellow. They're heading up the structural biology component of the QCRG. And an important point to make, again, Allison touched upon this, it's not just a collaboration between scientists here in the Bay Area and at UCSF and at QBI. It really was augmented to include researchers around the world. We've connected with over 25 different academic institutions. We've had some amazing industry relationships as well. And I'd say this is one of the themes I think of my seminar here today, and I'll come back at the end and go into a little bit more detail into it, is the realization how fast science can move when we actually collaborate. And I think the challenge now is to maybe keep some of this infrastructure and spirit alive as hopefully the pandemic dies down. It seems like we're still in near the beginning than the end, but as we transition and pivot back to studying other diseases, it would be great to keep this collaborative spirit in place. And again, I'll come back to that point at the very end of my talk. So here are a few papers that had came out last year from the QCRG. You can see they're incredibly collaborative. One, we initially generated a SARS-CoV-2 human protein-protein interaction map using an affinity tag purification mass spectrometry-based approach to identify human proteins that then hopefully could be targeted through drug repurposing. I'm going to speak a little bit about this. We're really focused a lot on the host response to infection. So we looked at changes with respect to phosphorylation in the host during the course of infection to try to find kinases that are most misregulated that then we could target pharmacologically. I'm not really going to go into details here to this study. I will highlight a few tidbits later on and show you how they weave into all the work that's ongoing. And then the third study is this very large collaborative study. It's a comparative host coronavirus protein interaction map revealing pan-viral disease mechanisms with the ultimate goal of trying to identify pan-viral disease therapeutic strategies. And it involves multiple different coronaviruses. But just a few words about this initial paper. So very early on in the pandemic, it was actually January 2020, David Gordon and Gwendolyn Zhang in my lab cloned out each one of these genes using the sequence that was reported. It was the first one reported actually in the United States from, I think it was in the state of Washington. And I think we were the first group to clone out each one of these genes. And our goal was to put affinity tags on them, express them in human cells, lyse the cells, purify the viral proteins, and then by mass spectrometry, identify the co-purifying human proteins so that we could generate hopefully a high quality SARS-CoV-2 human protein-protein interaction map. And this is something that we've been doing a lot of over the last 15 years, targeting a number of different pathogens, not just viruses, but bacterial species as well. We initially did this with HIV. We're still working on the mechanistic insights derived from this map here, this particular HIV human protein-protein interaction map, but also HPV, Ebola, fluomics. You can see there's, this is Priya Shah's great work focusing on dengue and Zika in human cells and mosquito cells, and then also looking at different bacterial systems, the secretome, if you will, tuberculosis and chlamydia. So we were in a perfect position to kind of pivot our pipeline to SARS-CoV-2 when it came online here at the beginning of 2020. So when we initially cloned out each one of these genes, we just simply tweeted from our account. I got to sneeze. No, it went away, sorry. So we tweeted out from our lab account that we had these clones. We're happy to ship them to whoever would want them. No strings attached, no MTAs. Please feel free to distribute. And in a matter of weeks, we actually sent these clones out to over 450 labs in over 40 countries. So to me, it was remarkable how fast we could get these genes out. I like to say that these plasmids or these constructs spread around the world a lot faster than the virus did. And we're very happy we were able to spread out these reagents so that we could expedite ultimately research on COVID-19 and SARS-CoV-2. So here is the initial iteration of the map that we had generated, where the red diamonds are the SARS-CoV-2 proteins, and then the other nodes are the human proteins. So in total, we had 332 human proteins. We gave this list to Brian Shoykat and Kayvon Shokak, two great chemical biologists here at QBI and UCSF. And what they did was then provide us with a list of 69 drugs or compounds that would bind to and inhibit at least one of the human proteins that we would hope that were highly enriched to be functionally relevant for the virus to be able to infect our cells. And we collaborated with scientists in New York and Paris and in other places, most notably Marco Vignuzzi at the Pasteur Institute and Adolfo Garcia Sastra at Mount Sinai Hospital in New York. They were able to test a lot of these compounds and drugs, and many of them did show potent antiviral effects. In total, when we look at all the studies that we've carried out here over the last year and a half, there's been 26 drugs that can be tied back to at least one of a piece of data from our work. We're monitoring all of these from afar. Some of them we're more intimately involved in. A couple of these clinical trials that I want to highlight. There's two translational inhibitors that are presently in clinical trials. The first one here is zodatifin. It's from the company Effector. This is a company that has generated drugs used to treat a variety of different cancers. And it binds to and inhibits a translational initiation protein. And this is a protein that is derived from our map. So we predicted that zodatifin could have antiviral effects. Sure enough, in cellular models and in the laboratory setting, it did. It's actually quite potent. And based on all that data, we partnered with DARPA to get $5 million and the company Effector to start a clinical trial with zodatifin. And this just started a couple months ago in 3 places in Florida, one in San Antonio and one at NIH. So we're excited about seeing the results of this particular clinical trial. The other drug that I want to highlight is another anti-cancer drug called Aplidin from the Spanish company PharmaMar. It also inhibits a translational protein, this time a translational elongation protein called EIF1A, and it's being used to treat multiple myeloma. And again, this is a connection with these translational apparatus that we uncovered in our protein-protein interaction map. So we predicted this could have potent antiviral effects. And this is work that I'm going to show you that was done collaboratively with Adolfo Garcia Sastra and Chris White at Mount Sinai. So here's the structure of Aplidin. It's called Aplidin when it's used to treat cancer, and it's being called plitidepsin when it's being used to treat COVID-19. Here's the structure of it. Interestingly, it comes exclusively out of this creature that lives off the coast of Ibiza, Spain. It's a sea squirt. And here's its target, like I said, EIF1A. And here's the structure of the target binding to Aplidin or plitidepsin. This isn't work from us. This was done previously. And we worked closely with Adolfo to show that it had very potent in vitro antiviral effects. So we've actually been involved in a lot of drug screening really around the world. And this particular compound or drug is the most potent that we've seen. It's actually got a sub nanomolar IC90 value. This is being done in HEC293 cells expressing ACE2. It's in this system 30 times more potent than remdesivir, very potent in primary lung cells, similar to remdesivir, but at a much lower concentration. And then with Adolfo, used one of his mouse models to see the efficacy in this particular species. So what he's got here is a mouse that with an adenovirus is expressing human ACE2. So then you can infect the mice with SARS-CoV-2. Then you add the drug, you extract the lungs, and then you look at the viral titers. And what you can see here, we're on the y-axis, we're looking essentially at the viral titers. You get about a 500-fold decrease of infectivity when you're using Aplidin, and much more potent than remdesivir, and again, at a much lower concentration. So in multiple models, it looks incredibly potent. And we've been communicating with the Spanish company PharmaMar all along. And based on our results, they'd actually had carried out a phase 1, phase 2 clinical trial, and very successful. This was just recently put up on medRxiv. And based on all of this information, there's actually a number of phase 3 clinical trials that are ongoing right now at 70 sites across 17 different countries. And we're actually in talks with India right now to see if this drug can get used there as well to augment the clinical trials that are ongoing. So this is another drug I think that we're very excited about, and we're looking forward to see the phase 3 clinical trial data soon. I mean, we have vaccines. This is great. We're clearly going to need more armament as we fight this SARS-CoV-2 and COVID-19. So hopefully you'll hear something positive about these two particular drugs. But just to highlight it, to me, they're particularly exciting because they were extracted from our protein-protein interaction map that we had generated now about 1.5 years ago. So then I want to segue to this paper here, this comparative host coronaviral protein-protein interaction analysis. And I find this one particularly exciting for a couple of reasons. One, it puts together, as you'll see, a lot of the different technologies that we and our collaborators have been using over the years. And as you can see, also, it's very collaborative. There's over 200 authors. There's 14 different institutions represented here from 6 different countries. And I would argue a work like this would probably normally take 5, 6, 7 years, and we did it in 5, 6 months. And that's a testament, in my mind, to the collaborative spirit that went into this particular work. And here's the overview of the study. So as I kind of referred to, we didn't just look at SARS-CoV-2, we looked at SARS-CoV-1 and MERS, cloned out each one of the genes from these species, did the same thing that we did for SARS-CoV-2, generated a protein-protein interaction map initially in HEC293 cells. In parallel, we're putting these proteins into cells one at a time and just carrying out microscopy with them to see in which subcellular organelle they were localizing to. For about half of the SARS-CoV-2 proteins, we actually generated antibodies for them, so we could do this localization in infected cells as well. The ultimate goal is to put all this information together to do kind of comparative network analysis, combine that with functional genetic validation experiments, focus in on key nodes that we think are important to carry out structural characterization of host pathogen interactions, and then ultimately try to combine all this information with real-world clinical data. So here is the SARS-CoV-1 human interaction map that we had generated, again, using the same pipeline that I had outlined previously and Apriya had used for dengue and Zika. So here we're looking at 25 SARS-CoV-1 proteins connected to 365 human proteins. Here's the corresponding MERS map, 22 MERS proteins connected to 292 human proteins. And there's a number of different ways you can compare across these data sets. A lot of sophisticated ways. We've been working with this type of analysis with Trey Eidecker over the last decade or so, kind of doing these cross-comparative studies. But one very simple way to just get an overview of the differences and similarities is just simply hierarchical clustering. Okay, so here's just a clustering of the data. And what this reveals is about 1/5 of the interactions you can see in all 3 viruses. About 1/4 you see exclusively with SARS-1 and SARS-2, and then about 1/5 that are MERS specific. So there's some similarities, there's some differences. And we and many others are looking at all of this data in this fashion and extracting out, I think, very interesting mechanistic insight about how coronaviruses come in and infect and revirus our cells, and ultimately how coronaviruses evolve as well. So I encourage you to look at this information if you're interested in these areas. So as I said, what we're also doing here is looking via localization, microscopy, just doing transfections of all individual proteins into cells and seeing where they localize to. A lot of this has been done with Robert and Svenja at Freiburg and with Andrew in Sheffield. And then, as I alluded to earlier, 14 of the SARS-CoV-2 proteins we actually have antibodies for. Massimo Pellemerini from Glasgow generated these so that we could do the localization in infected cells as well. So we generated a lot of images from this analysis. And of course, all of this is out there for the public to look at. But what we did initially for this paper was try to distill all this information down into some sort of digestible tables. And this is what we tried to do here, kind of summarizing this data, where you have the 3 different viruses here on this axis, you have the different viral proteins, and then this is where we think that the different viral proteins are localizing to based on the microscopy experiments. And we could then lay on top with about half of the SARS-CoV-2 proteins localization information from infected cells. And this is what we've done here, looking at strong, good, and weak localization laid on top. So you can compare the blue boxes, which are from the transient transfections, to these other colors from infected. And you can see some overlap, but there's some differences as well. It's a lot of information here, but I want to highlight one particular connection, and it has to do with the one protein, ORF9b, which doesn't exist in MERS, which does in SARS-1 and SARS-2. And you can see that it's localizing to the mitochondria here with the SARS-CoV-1 ORF9b, as well as with the SARS-CoV-2 ORF9b, and using an antibody against ORF9b. And this is the localization image from the antibody against ORF9b in cells infected with SARS-CoV-2. And this is a quintessential mitochondrial pattern. So I just want you to remember that ORF9b localizes to the mitochondria, and because I'm going to come back to that in just a couple of minutes. So also in parallel, as I had outlined in the overview, we were looking at the interacting proteins and systematically perturbing them genetically to see what effects they have on infection. And we're doing this in a couple of different ways. One, we worked with Marco and Bronico at the Pasteur Institute, where they were doing RNAi knockdowns we started with the 332 human interacting proteins with SARS-CoV-2. We knocked them down in A549 cells expressing ACE2 using an RT-PCR readout, looking as a proxy for infectivity. In parallel, we actually collaborated with this company in South San Francisco, Synthego. This is a great example of the kind of industry relationships that had been formed over the pandemic. We work closely with these guys, which are really very enthusiastic, Jared and Kevin. And what they did was knock out each one of these genes one at a time using CRISPR and CACO2 cells. They were then sent off to Chris Bassler at Georgia State University in Atlanta. And then he carried out this following assay for us, where he took the knockouts and then infected. And he has a different assay here where you're taking the virus off the cells and then reinfecting other cells. And here he's using an antibody against N as his readout for infectivity. So we had two different genetic perturbation strategies and two different cell types, two different assays in two different parts of the world to kind of give us an overview of how many of these interacting proteins were actually functionally relevant. And in total, based on both of these pipelines, we identified 73 out of the 332. And this is a summary of both data. The red nodes here are genes that when knocked out with RNAi that had an effect. The ones in yellow are the ones that when knocked out in CACO2 cells have an effect. And then the ones in orange, you see have an effect in both systems. So there's, again, some overlap and there's some differences. But overall, we identified 73 out of these genes having a functionally relevant effect. And here's a representation, another representation of those 73 genes with respect to which SARS-CoV-2 or SARS-CoV-1 or MERS proteins that they physically interact with. So the blue nodes are the 73 genes, and then the edges correspond to physical connections with proteins from SARS-CoV-2 in red, SARS-CoV-1 in orange, and then MERS in yellow. I want to zoom up on one particular connection that has to do with ORF9b. So now I'm coming back to ORF9b. So we found that ORF9b from SARS-CoV-1 and SARS-CoV-2 physically interacts with TOM70, and the knockout of TOM70 in CACO2 cells results in an order of magnitude decrease of infectivity, right? So we have multiple lines of evidence to suggest TOM70 in this particular connection could have great value to go look at more mechanistically. And these are the exact kind of connections that then we were handing off to the Structural Biology Consortium in the QCRG, as I had referred to at the beginning of my talk, led by Oren Rosenberg and Clem Verba. They got together 60 different researchers, mostly trainees, students, and postdocs from 18 different labs, and set up this amazing infrastructure, these different pipelines focused on protein expression or crystallography or cryo-EM or computational approaches to kind of study these proteins structurally. So they've been marching through a number of these interesting connections, including this connection between TOM70 and ORF9b. So they were able to get a 3-angstrom cryo-EM structure of this interaction. ORF9b is in orange here, TOM70 in green. So what we're seeing here is that ORF9b is being tucked up in the active site of TOM70. And the question is, okay, what is TOM70? So I referred to the fact that it's mitochondrial, and what it does is import, export proteins in and out of the mitochondria. So one hypothesis here is that, all right, maybe ORF9b is using it to get itself in mitochondria to wreak havoc during the course of infection, or maybe it's also impeding other human proteins from getting in or out of the mitochondria in a way that's beneficial for the virus. So we're looking into that. But what also TOM70 does is regulates the immune response. And we think that ORF9b could be manipulating both of these pathways, but we've been focused a lot on this latter pathway, especially now as we've been focusing on variants, which I'll come to in just a few minutes. So I just want you to remember that connection as well. Here's a few more just tidbits about this particular structure. The interaction that we uncovered is mostly hydrophobic, with 36 out of the 38 ORF9b residues interacting with TOM70. And very interestingly, through our phosphorylation work, which I didn't have time to go through, this was led by a postdoc, Medi here at UCSF, we found that 2 residues on ORF9b at S50 and S53 are actually phosphorylated. And those are residues that are tucked up here in the active site. And the hypothesis that we had initially was, well, if these residues were phosphorylated, biophysically, there's no way ORF9b could be interacting with Tom70. And that was something that we tested here in a subsequent study, and I'll come back to that. I just want to say a few more words about ORF9b. I find it incredibly fascinating. We call it the shapeshifter because it takes on these different shapes depending on its context. So when it's by itself, it forms this dimer, and it's primarily this beta sheet configuration. But when it's a monomer binding to Tom70, it has a very different configuration. You see this alpha helices. And this is quite rare for proteins to behave so differently depending on who they're interacting with. And this could be one of the reasons why SARS-CoV-2 proteins and maybe viral proteins in general can have such a big effect with such a limited genomic capacity. Maybe the individual proteins are basically having different shapes depending on who they're talking to. So this is something we're looking at more systematically, especially with SARS-CoV-2. And I also really like this example because it illustrates the power of putting together proteomics and biochemistry and genetics and virology and structural biology together to come up with a key node, which we think is important, not just for SARS-CoV-2, but SARS-CoV-1 and presumably SARS-CoV-3 when we ultimately have to encounter it. So we think this is a great drug target. We're working with Brian Shoykat and James Frazier here to do some fragment-based screening to try to find some chemical matter that would try to interrupt this particular interaction. Now, I want to tell you one other quick vignette here that came out of the QCRG Structural Biology Consortium. It has to do with combining cryo-EM with AI. And I'm sure everybody's very well familiar with the huge breakthroughs in AI and AlphaFold and DeepMind and Google recently. Well, they'd been trying to get a cryo-EM structure of NSP2. Its function is pretty much unknown. It's a very conserved corneal proteins, but not much is known about what it does. And what they were able to do, Klim and Oren and Adam and Megna and many, many others, they were able to get about a 3-angstrom structure, about the 70% N-terminal end of SARS-CoV-2. But this last piece here was very blurry. They couldn't get it, although it's resolved here, but at the time, they couldn't get a resolution. Well, ironically, at the time that they were kind of describing, Google was, what they were doing with AlphaFold, they had actually just put out the 5 proteins that they randomly seemingly selected and said, oh, can we get a structure of these? And they actually tried NSP2 and they just put it on their website. So this was several months ago before this paper was published. And Klim found it and he said, well, look at the structure. Like all the individual domains are right, but take a step back. The configuration as a whole was wrong. But he said, I'm going to take this information from AI, put it with this cryo-EM, and this is how we could get a final structure of NSP2. And then when we go back and look at our NSP2 human protein-protein interaction map, we find here that NSP2 interacts with this one protein, RAP1 GDS1, another kind of uncharacterized protein in the host that works upstream of a number of GTPases. And they were able to get a cryo-EM structure, albeit a lower resolution cryo-EM structure of NSP2 binding to RAP1 GDS1. And then, of course, we get these structures. One of the goals now is to work with the great chemical biologists here to go ultimately to structure-based drug design. And I just want to, I really like this example because I believe it's one of kind of the future threads of structural biology. I don't think AI is going to get you all the way there, but it's say if you had cryo-EM or tomography getting you partially the way there, I think AI can come in and really clean things up. So we're really looking to push a merging kind of cryo-EM and tomography with AI here at QBI and QCRG and ultimately with at UCSF as well. And this is a paper that's out on bioRxiv if you're interested in learning more about it. So I'm going to end now by talking about some of our work on the variants. These are the problematic viruses that are making us Zoom these seminars. Hopefully we can get back in person soon, and we have to deal with these variants though in order to make that happen. So we've been looking at these 4 variants, alpha, beta, gamma, delta. We started looking at the variants when the alpha was the most problematic. And now, of course, we're focusing a lot more attention on delta. But I want to tell you a little bit about our work on the alpha variant because I think it does pertain to the other variants, especially delta as well. Here's the B117, the alpha variant. And here it is compared to 2 earlier lineage viruses that we're also been looking at. IC19, this is a virus that was one of the initial ones identified in England at the start of the pandemic. And then Vic here stands for Victoria from Australia. Again, this is another virus that is tracked back to early on in the pandemic. So we did some comparative studies comparing the Alpha variant to these two earlier lineage variants. The first thing that we did, and I should say this has all been done in great collaboration with Greg Towers and Claire Jolly at University College London. They were one of the first groups in the world to get these variants. We were comparing across all these three and just looking at intracellular viral RNA, we actually didn't see a big effect. And then looking at the infectious virions that came off the infected cells, how infectious were they? And we actually didn't see such a big effect, which is kind of a surprise looking across these three different variants. But then what we wanted to do was use some of our unbiased tools that we've been working on, not just with pathogenesis, but other disease areas over the last 10, 15 years, to see if we could get some insight behind why Alpha variant was being more problematic in these earlier lineage viruses. So to this effect, we took the three viruses, infected cells, and did temporally analysis at 10 hours and 24 hours post-infection, did RNA-seq, mass spectrometry, abundance proteomics, and phosphoproteomics to see if we could tease out why this Alpha variant was so much more problematic. And what we saw here when we were globally looking at all this data and comparing, again, the data we're getting from the Alpha variant from the earlier lineage viruses, that the pathways that were most differentially perturbed or manipulated with the Alpha variant were those that were connected to the immune response. So this is the top 5 categories that were manipulated using RNA-seq. You can see there's interferon signaling that's perturbed. You can also see that when you look at abundance proteomics, and then you look at the phosphoproteomics as well, you can see that proteins that are connected to the immune system are the ones that are being differentially phosphorylated. And just to show you a little bit of this data, I'm just going to show you the RNA-seq data, and I'm specifically showing you the ISGs, the interferon stimulated genes. Those are the genes that are turned up when you get infected, and you need these to be turned up to fight off infection. So we're looking at 10 hours and 24 hours post-infection, comparing the Alpha variant to these two earlier lineage viruses. And what you can see, I think quite strongly, is that you don't get these genes turned up when you're using the Alpha variant. So there seems to be a suppression of the immune response with respect to the Alpha variant compared to the other viruses. And of course, that would be very beneficial for the virus, because if you don't turn on these genes, you can't fight off infection, you get more virus, and ultimately that could contribute to increased transmissibility and ultimately increased mortality. So that was one trend that we uncovered using these unbiased approaches, looking at the host. Then we wanted to look at the genes and the proteins corresponding to the virus. And what we saw here very strikingly was ORF9b, both by RNA-seq and by proteomics, is very turned up with cells infected by the Alpha variant compared to these older lineage viruses. So I showed you 2 pieces of data here, right? Is that the Alpha variant seemingly is not allowing or suppressing the immune response, and 2, ORF9b seemingly is turned up a lot higher with the Alpha variant compared to the other viruses. And the question is there some connection between these two? Well, before I get into that, I just also want to tell you that although we were doing this in cells in a laboratory setting, there's this paper that came out of bioRxiv showing that ORF9b is also elevated in patients infected with the Alpha variant, which was really exciting to see. What they hypothesized here is that maybe that ORF9b is being turned up through one of the mutations in the N protein. So N and ORF9b are overlapping. So there's a mutation in the 3rd amino acid of N, changes it from a D to an L. But this mutation also manipulates the sequence just upstream of the ATG start codon for ORF9b. And they hypothesized maybe this manipulation somehow is misregulating ORF9b, so you get more ORF9b. This is something that we're looking at as well. We haven't definitively shown that. There's some other mutations we're looking at as, in addition. But nonetheless, it was nice to see the phenomenon that we saw in cells in the lab also exist in human situation. So ORF9b is an old friend, right? We talked about this before. We find ORF9b interacting with TOM70, our protein-protein interaction analysis. We find that ORF9b is phosphorylated on these 2 residues, as I talked about before. We find that ORF9b from SARS-1 and SARS-2 interacts with TOM70. When you knock out TOM70, infection goes down significantly, and then we got a cryo-EM structure of TOM70 binding to ORF9b. So we had a lot of connections to ORF9b as this particular connection became apparent when we were starting to study the Alpha variant. So I want to go back now to this particular hypothesis that I had postulated before, is that if the ORF9b was phosphorylated on serine 50 and serine 53, we would say that it could no longer interact with TOM70. And Jody Batra, a scientist in the lab, carried out the following experiments for us, where she made phosphomimics at these two residues. And she showed, sure enough, when you have an S53 to an E or a double mutant here, that ORF9b no longer binds to TOM70. Okay, so phosphorylation would impede the interaction, seemingly, between ORF9b and TOM70. So I told you before that TOM70 is connected to regulating the immune response. And there's been a couple papers now in the literature showing that ORF9b also manipulates the interferon response. These are two papers here. And in this particular case, there's a hypothesis really that ORF9b is suppressing type 1 interferon responses through its interaction with TOM70. Okay, so this I think is a really key piece of data for this particular paper. And we actually showed that ORF9b itself, when you put it into cells, suppresses the innate immune response. So here's an assay where we're essentially looking at the immune response. It's a luciferase assay that's on this axis, and you're adding poly-IC, which can induce the immune response. And here's the negative control empty vector. This is the response you'd expect to see. Now, when you do this experiment, when you overexpress wild-type ORF9b, you can see you get pretty good suppression. But importantly, when you do it in the presence of ORF9b that's mutated in a way that can no longer interact with TOM70, you do not get that suppression. So we think that the alpha variant is suppressing the immune response through ORF9b, through its interaction ultimately with TOM70. So here's our working model. So here's normally what happens in the course of infection. Okay, you get the RNA and an ORF9b are overlapping, as I pointed out. There's some ORF9b obviously being expressed with the earlier lineage viruses, binds to TOM70. This is how you get a reduction of the immune response, and this is how one can facilitate, or the virus can facilitate, transmission. But what's happening here with the alpha variant, that there's mutations outside of spike that are seemingly having an effect. This is one of our hypothesized mutants that's responsible for this. We have other ones as well. But there's mutants here that are given as more ORF9b, and the more ORF9b there is, the more combined to TOM70, and the more strong the suppression of the immune response can be. And that's how you get ultimately increased transmission and indirectly increased mortality. So this is a model that we're highlighting, and I think it's important for a couple of reasons. It's saying, hey, there's mutations outside of spike that are important. It seems like the whole world is just focused on spike. Well, let's look at some of these other mutations. And to me, it's remarkable about how it seems like SARS-CoV-2 is manipulating our immune response after it gets into cells in a way that's incredibly beneficial. And this phenomenon that I'm describing, we're looking at it across the other variants. And very excitingly, we're seeing similar patterns as we go from variant to variant. Maybe not the same mutations, but the end result seemingly looks like it's the same. And this comes just a couple of concluding slides on that front. As I said, not just alpha, we're looking at all the variants. And we're doing this a variety of different ways: phosphoproteomics, abundance proteomics, ubiquitination proteomics, drug screening, CRISPR-based analysis, RNA-seq. We actually have cells now expressing human ACE2. This was work done by Jody Batra. We can actually infect these cells with SARS-CoV-2. So we can carry out a whole slew of experiments in the bat cells with SARS-CoV-2 to try to get at potentially mechanism behind the putative zoonotic event that happened between bats and humans. So we're doing all of this in the context of infection, but we're also doing this looking at the individual mutations as well. So we've synthesized all the individual mutants with all of these different variants. We're looking at what effects the mutations have on protein-protein interactions, doing biochemistry with QCRG were relevant, structural analysis were relevant. This is ORF8 structure. Again, looking at these in bat cells, post-translational modification mapping like we've done before, and then genetic analysis using CRISPR and/or RNAi, and also in collaboration with a number of great virologists, making pseudoviruses to see what effects the individual mutations have. And then ultimately, as we've done, is distribute all of these mutated reagents to the community as well. So there's a lot of work to do to get at the underlying biology of why these variants are seemingly so much more problematic than some of these earlier lineage viruses. And just to wrap up, I had mentioned this earlier on in my seminar. I want to come back to this point about collaboration during the pandemic. And I think everybody has been able to really nicely see when boundaries are broken down, silos are busted, when people come together from different disciplines, different academic institutions, between pharmaceutical companies and the academic world, look how fast we can move, right? This to me has been absolutely remarkable. And why hasn't this happened before as we study other diseases in the past? And I think there's problems with the academic system in general in that collaboration is often very discouraged. You know, who gets the awards, who gets tenure, who gets a grant? An individual. And oftentimes when you have these larger collaborative groups, people say, well, what did that person do in this collaboration? You know, why? I want to see a 2-author paper. And I couldn't disagree more with that type of logic, because you can see, look how fast you can move when we all work together. So my hope is that this collaborative spirit and this infrastructure stays in place as hopefully this pandemic dies down, so that we can focus on all disease like this. Why aren't we doing this type of work on breast cancer and Alzheimer's and Parkinson's? So the hope is that we can in the future. And my optimistic hope is that 20 years from now, we'll look back at this pandemic and obviously realize it's a tragedy. But I think we're going to have a lot more treatments for a lot of other diseases, because we learned so much, not just about science, but how to work together and how to accomplish things. So that's hopefully my positive message here at the end of the day. So this was a piece that Cell asked us to write. I wrote this with Jacqueline Fabius, who's the Chief Operating Officer of the Quantitative Biosciences Institute. And then Nature recently highlighted our efforts, as well as many other collaborative efforts during the pandemic. So I'd encourage you to read these pieces if you'd like to know more about the type of things we have done and the type of things that we ultimately want to do. So obviously a lot of people around the world that we've worked with, I hope they have done justice in recognizing them. This is a picture of my lab that was taken a few months before the pandemic, seems like many, many years ago now, but a few people to highlight here. David Gordon and Gwendolyn Zhang, as I said, were really the first to kind of spearhead our protein-protein interaction analysis on SARS-CoV-2. Mehdi, computational biologist, he's experimentalist too. I should give him credit for that too. He spearheaded our phosphorylation work, and computationally has been done kind of the foundation for a lot of our SARS-CoV-2 work. And then Robin Cake, Ruth Hittenhain, and Daniel Sweeney. These are 3 fantastic mass spectrometrists that were heavily involved in these projects, and actually on all the projects that we're involved in. There's such great people to work with. So I'd like to thank them for all their hard work. And finally, I'd like to thank you all for your attention. I'd be happy to take questions now.
Thank you, Nevan. That was fantastic. And certainly your tribute to interactive science and connectivity among scientists, big science, is something that resonates here, I think, at UC Davis. We have some questions in the chat box. To kick off, Angela asks the basic question, what do you mean by the innate immune response?
Where is that question here?
It's there, right there on the chat box. I'm reading
Well,
directly from. What do you think of adaptive clinical trials, and what would be your list for one?
So those are 2 questions there, right, Ralph? Were those
Yeah,
2?
Two very different questions.
Yeah, so
the same person. They're both from Angela.
So the what we're looking at here obviously is a molecular readout, proteins and expression of genes, looking at the ISGs. And then the hope that the kind of mechanisms we're uncovering are similar in ultimately in mice and in humans. And the fact that we're seeing at least some preliminary data suggesting that some of the phenomenon that we're uncovering at a molecular level in the cellular systems are seemingly being recapitulated in the humans. And we're getting more into those areas in collaboration with a number of scientists, I guess, around the world. So right now, what we can do is focus on the molecular readouts of the immune response. I hope that answered your question.
No, actually, my question was exactly what's the molecular readout of the immune response? Because you just showed immune suppression or immune enhancement, but I was just curious.
Well, we were looking, so we look via RNA-seq, we're looking at all the genes. By abundance proteomics, we're looking at, in theory, all the proteins, but we don't obviously identify them all. And then we're looking globally at the phosphorylation changes as well on those proteins. And we've done this in cells infected for the alpha variant, in other variants, comparing this to earlier lineage viruses. And we're saying, okay, what's different? What's molecularly different between a variant of interest or a variant of concern with some of these earlier lineage viruses? And incredibly strongly, in an unbiased way, is genes connected to the immune response, to in particular the ISGs. So that, in a very unbiased way, really allowed us to focus on, all right, there's something here. And then we looked at it in more mechanistic detail, collaboratively with Greg Towers and Claire Jolly, to show that ORF9b looks like, through TOM70, is manipulating these pathways. And as I showed you in human cell, in humans, they see a similar phenomenon in terms of orphan being turned up as well with the alpha variant.
So Nevan, if I can just take you off a little bit on that tangent of the innate immune response. I mean, you are doing experiments now on bat cells, and the intriguing question, of course, is why these viruses, the coronaviruses, seem to be able to coexist quite peacefully within the milieu of the bat cell. And the question has come up about dampening of the bat immune response. How do you see that? For example, NLRP3, I believe, is one of the ones that has been incriminated in terms of saying the bat has that switched off.
Yeah, I think studying bats is just going to be absolutely fascinating. And I hope NIH realizes the value of studying this work in bats. It's been hard to get some money to do that in bats because it's not humans. But I mean, in order to study this, viruses, you got to study in the context of evolution, almost certainly. But a couple of things I'll say about the bat cells. I mean, we actually just got this data a couple of days ago. We generated a SARS-CoV-2 bat protein-protein interaction map. There's some similarities, there's some differences, almost certainly in these differences are kind of getting to what you're at after here, Ralph. There's a number of immune connections with these differences, and we got to go deep kind of in that regard. One of the things that we're gearing up to do is a CRISPR screen, right? A bat CRISPR screen, which I guess hasn't been done before, looking for genes that when perturbed either perturb infection or enhance it. And I think the ones that enhance it are going to be the ones that are going to have kind of these restriction factors that may be so important as we look at how the virus jumps from species to species. And the last thing I want to say too, which I think is I'm very interested in looking at, is ORF8. So if you look at ORF8, it never gets deleted when it's in bats. It often gets deleted when it gets into humans. So we have an ORF8 Delta virus, and I'm very, very excited and interested to see how it behaves in the bat cells compared to the human cells. I personally think ORF8 is going to be a key node here as we study how bats and humans behave differently. The last thing I just say, when you actually sequence bats, you see so many viruses in there. The question is, how do they coexist in these cells, right? Isn't that fascinating? So not just retroviruses, but a lot of these other viruses that are seemingly swimming around. So I think there's so much work to do in the bat cells. You can tell I'm very excited about bats.
Yeah. So Priya's asking the question, what keeps you up at night in terms of viruses and chronic diseases, apart from COVID? You know, what's the connection there? And do you, is that a cause of your insomnia, scientific insomnia?
Well, I hadn't thought of that, Priya, but maybe that is one of the reasons why I don't sleep that much. You know, I guess the kind of the long COVID symptoms and more around the kind of neurological side, I'm actually quite interested and fascinated with. I mean, Priya, it's similar to the work that you did, I guess, with Zika, connecting it to microcephaly through these kind of approaches. We're starting to generate these maps now in inhibitory neurons, excitatory neurons, NPCs. I'm very interested to see what, you know, how this data is connected ultimately to the brain, because I think the brain issues are very integral to the long COVID symptoms that are being uncovered. So I think this virus, it just gets in so many organs. So it's scary, but then it's a lot of work for us to do, because I think each organ represents kind of its own project, if you will, and understanding how it infects these different organs, I think will ultimately shed light on long COVID.
So Satya Dandekar has a few questions, but would basically like to know, are there interactions like the ones you described for ORF9b and TOM70 in other, in mucosal cells in terms of other viral pathogens? Is this a sort of model for mitochondrial connection, if I'm reading correctly into your
No, that's a great question. And I'm glad I showed that one slide with all our host pathogen interactions, or a lot of the maps that we've generated. What we're doing now is retrospectively saying, all right, what are some commonalities starting with SARS-CoV-2? And actually, when you look at which host protein, you look at the host proteins that are most represented across all these maps, TOM70 is like number 3. So it's in seven of these maps. So then you go back and look at the pathogenic protein and using AI, like, so maybe the primary amino acid sequence, you're like, well, there's no overlap here. But then if you can start to make predictions about structures, we're starting to see similarities at the structural level across these proteins. I mean, how cool is that? Now we're starting to do biochemistry on a number of these other proteins. We actually got one that's just reconstituted. We'll have a cryo-EM structure of it, I think, very soon, of a completely seemingly different viral protein from a completely different virus that binds to TOM70. And the theory would be, all right, maybe there's a similar mechanism at play here. So to me, that's such an amazing direction to be going now that we have all these data sets in hand. So that's a very timely question right now, and one that's getting us very excited as we go across, other proteins as well across these different pathogens.
So Bill Murphy's got a question. It's a long one. I'm going to read it so that I don't really, so that I can capture it exactly the way he's phrasing it. So one thing he says that's seriously lacking is the lack of an in vivo preclinical experimental data set on these approaches on overall immune responses, both short and long term. Many of the TKIs and other cancer drugs are now known to also mediate immune effects. How are these going to be dissected as innate and adaptive immune responses are dynamic and impacted by age?
I mean, I'm sure these are great points. More data collected from humans in this regard. I mean, we're starting to gear up to do this type of thing in collaboration with a number of groups, really, a lot of groups in New York. So these are very important questions and comments. And I think many of us, most of the scientific world is really at its infancy in terms of collecting the data that would actually address these concerns and comments. But we're trying really hard to kind of translate our molecular findings into, let's say, patients and trying to merge these two worlds. So those are our efforts on those fronts.
And what about tyrosine kinase inhibitors and how they connect with all of this? Is that just a class that is a part of the subset, or how do you view that?
Yeah, I mean, it's a good point. We're focusing, based on our phosphorylation studies, we're focusing on a number of different kinase families. The P38 is the one that we're most excited about. We're actually collaborating with 2 companies on clinical trials on that. That came out of some of our studies. So the tyrosine kinase work, we haven't focused on for the main reason it didn't come out unbiasedly through the work that we've been looking at. But hopefully soon we will be able to focus on those.
Paul Usu has a question. Comments on ORF9b shape-shifting in relation to prion and prion-like proteins.
Yeah, that's a great point. I got Stanley Prusner just down on the other side of the quad here. I was trying to find a time to talk to him about this, right? I think that's a great point. Are there similarities here, right? At some level with the prion proteins, maybe even just at the biophysical or biochemical or structural level. So I think this is a very great point, and this is something that's been in my mind for a while that we need to really take a hard look at.
Well, I think I've covered all the questions that have appeared in the chat box. If anybody else has a burning question, either raise your hand or follow up on some of the, Sacha, I think, has a follow-up.
Yes.
Yeah.
Dr. Krogan, this is absolutely fantastic presentation. We really enjoyed. Thanks for taking your time and doing this. I'm still coming back to your ORF9b. If you add to the cells, would it enter the mitochondria and affect you are seeing, you would see it? Is that possible? I was just wondering how it's getting localized to mitochondria.
That's a great question. Initially, when we saw this connection, we started to focus a lot of our attention on mitochondria. We still are, but then realizing that TOM70 is also regulating ERF3, which is a transcription factor that regulates the ISGs. We really shifted our focus there. We're going back to the mitochondria. There has to be something there, right, with the mitochondrial connection. And there's some ongoing work, tomography work. We're looking at in cells, what's happening to the mitochondria in infected situation or just overexpressing ORF9b. So there's a number of mitochondrial readouts that we're looking at now. So, I mean, these proteins are very sneaky, right? They're going to have multiple roles in multiple different processes. So almost certainly ORF9b is manipulating the mitochondria as well. in a way that is beneficial for the virus. So we just haven't uncovered that, but we're looking at it. We're looking at that connection.
Has Tom70 been seen in other viral infections which affect the mucosal sites? Have you seen that in your interaction maps?
Well, we've got Tom70 out of these other host pathogen interaction maps that I referred to, but I don't know. Beyond that, I mean, we really need to take a hard look at that. We've been focusing a lot on the biochemistry and the biophysics and the structure now, but we have to take a few steps back and say, you know, what is linked with Tom70 across many different data sets. There's got to be a lot more there.
Thank you.
So, hi, this is Bill. That was very interesting. You know, I was at NCI for many years, and they had the NCI 60 panel of cell lines, and they did the similar kind of drug discovery, you know, in the 70s and 80s. One thing that they found was you still had to, you can't go to the clinical to dissect the immune aspects. You know, the bottom line is it's just too variable, too many, you're not going to get any kind of mechanism, and it's going to be very time consuming. The question is, in this kind of schema that you have, which is excellent, the preclinical, I mean, as far as then looking at the long term, because ultimately the immune, and everybody's talking about combining an immune attack with targeted agents, how is that going to be, you know, assessed? Because you can't go from the test tube right into let's try it in people and then figure out how to optimize, because ultimately it's going to be the immune response that will give you long-lasting protection, right? And then is it impaired by some of these, just like with the old HIV drugs? You know, you have to keep those kinds of caveats. I guess that's what I'd like to see is how is that being mobilized? Because I don't see, at least with the models that we have, or even piggybacking with other viruses, how we're going to build on some of these, like, you know, existing drugs or even new ones on all those different immune parameters. That there's a niche there that I think is just sort of a disconnect.
Yeah, and look, I completely agree with that. And this comes back to kind of the overarching theme is this disconnect that you refer to. Hopefully we can connect it, and then it will have a huge impact, not just on this, but on so many other things in the future, right? So, I mean, so a couple of things I'd say is, I mean, I guess what we're doing, kind of the preclinical, I mean, there's mouse models we're using, there's ferret models, there's hamsters models, you know. So in that regard with drugs, we have those, but then kind of just understanding the immune response. What we do for a living is unbiasedly collect data. So what we need to do is collect in a more non-biased way information from humans, and then we got this other data, and then say, hey, where's the connection? And how do we integrate this information together? So we're really, as I was kind of alluding to earlier, really at the infancy in terms of collecting that information. But you bring up such a good point, because I mean, that's really, in my mind, what this pandemic has done is allowed us to identify where the disconnects are.
Yeah.
Right. And then, all right, we got to shore those up. And out of fear, it's just that we got to shore these up. And then what we've learned here to make these bridges and to break down these silos, let's apply these across all these other disease areas. That's why overall, I'm quite optimistic of the kind of the research community and the biomedical world to really, we're learning so much on so many levels, and the challenge is now to apply it to all these other disease areas.
Yeah, no, thank you. I guess because UC Davis has so many of the models, and it's just that, you know, then you can look long term. The long COVID is an excellent example.
Yes.
Right. And then also some of the vaccine effects, you know, where you need to be looking at some of these. So I think in that sense, the models are here. It's just that, you know, getting them tapped into would have been nice, looking at longer term immune.
Well, especially you have these great monkey models there. I mean, we should be working together on this, right? Like just
I agree.
Getting some information, and we could hopefully find some, in a relatively short period of time, some profound connections that could have impact.
That's what these sessions are all about. Look, the hour is kind of winding down. This could clearly go on for a long time, but I want to hand back to either Prasant or Allison to wrap this up. And again, Nevan, thank you so much.
Thank you.
So thank you so much, Ralph, and thank you, Nevan. This has been an amazing talk, and so very valuable. I'm having trouble getting my video back on, so I apologize. But it's been a tremendous time here to really think creatively, and I love your focus on teamwork and the whole team. It's really, you know, that's one of the things we've learned so much about the pandemic through all of this is the emphasis on teamwork and collaboration and interdisciplinary research. And your work really epitomizes that. And we're so pleased that you were able to share this with us. And this is a pivotal time in medicine across the country. And you mentioned something, we wouldn't have been able to do that before. And you've shown that, and so has UC Davis shown that on really how to make a difference by bringing everyone together. So we're so appreciative. And I just want to thank the team, Ana Lucia and others who really, and especially Ralph, who always is bringing his friends to bear and sharing great information with us. Thank you again.
Thanks, everybody. It was a pleasure.